Again, welcome back to MLT 120. Today we're going to talk about mononuclear white blood cells. These include our lymphocytes and our monocytes. We're going to first focus on our lymphocyte origin. The primary lymphoid organs are our bone marrow and our thymus. Primary means the lymphs develop and mature there. Secondary lymph organs include our spleen, our lymph nodes, and our tonsils. This is where the lymphs reside after they mature. The path our lymphocytes can travel, they can either travel between areas of inflammation, or they can move from the bone marrow to the thymus or into our secondary tissues of the lymphatic system. If you look over here, here's a perfect example of what our lymphocyte looks like. Remember, we used our lymphocyte as our guide or our gauge to determine the size of our red blood cells when we're trying to determine whether our red blood cells are too big or too small. And we were looking at the nucleus of our lymphocyte. So that's the only really contact we've had with the lymphocyte so far, is just using it as a gauge. But usually our lymphocytes are, unless they're active, they're nice and round. They have a nice round to oval nucleus, and the nucleus should be roughly the same size as a normal sized red blood cell. And of course, if the red blood cells are not roughly the same size, then the red blood cells are either microcytic or macrocytic. So we have 3 steps in the maturation sequence for our lymphocytes, and you can look at these in your atlas to see a little bit better images. But we have our lymphoblast on page 81 in the atlas, and we have our prolymphocyte as stage 2, and then we have the mature lymphocyte. And these can mature into our T cells, our B cells, or our natural killer cells. And the T cells and B cells are involved in the adaptive immunity, which involves recognition, memory of foreign invaders, and antibody production, whereas the natural killer cells are part of the innate immunity, which is our everyday protection. No memory or antibody production is involved. And remember, this whole line is coming down on that lymphoid line. Remember, we start out with the hematopoietic stem cell, or the pluripotential stem cell, and then it splits off to myeloid and lymphoid. This whole line of maturation is on that lymphoid side, which we haven't really talked about to this point. So these are all on that lymphoid side. And you should notice something else. We've been talking about granulocytes. These mononuclear cells do not have those granules in their cytoplasm like our granulocytes, our other white blood cells that we've been talking about. So our monocytes do not have those granules in the cytoplasm like our granulocytes. So our lymphocytes can either be T cells or B cells. So let's talk about T cells first, because T cells are about 80% of circulating lymphs in the peripheral blood. They are responsible for cell-mediated immunity, or recognition of self from non-self. They originate in the thymus, which is found in the upper chest area. So here is an image of the thymus. And they're long-lived. They can live months, and they can live up to about 6 months usually, the T cells. So now let's talk about a couple of subpopulations of our T cells. First off, to understand these subpopulations, we need to understand what CD is representing. CD is a cluster destination, which is just surface markers on the lymphs themselves. So anytime you see the CD, it's just referring to cluster destinations, and it's how these subpopulations of cells are named because of the markers on their surface. So the first subpopulation is CD4, and these are helper cells. And that's what these couple of images up here are talking about or showing. But the helper cells interact with macrophages, and this is a macrophage right here, and the macrophages come from monocytes. And they secrete cytokines and promote humoral immunity. And humoral immunity involves the production of antibodies. And if your body is building up antibodies, it's building up immunity. So the helper cells and the macrophages are working together for your body's immunity. Just for note, if you're an HIV patient, the HIV infects the T helper cells and it disables that person's immunity. So HIV, the people that have HIV, they have a real problem with their body being able to protect themselves. Over here in this image, helper cells also interact with B cells. So again, T helper, they're helping other cells in the body's immunity. The second cluster destination or subpopulation of T cells is the CD8, which are T cytotoxic or suppressor cells. These are cytotoxic, eliminating non-self suppressor antibody production, and these basically helps turn off the immune response when it's no longer needed. Okay, the body is really good about protecting itself, but sometimes there comes a point when the immune response is no longer needed. So that's kind of what these CD8 T cells do. They help turn off that immune response. Okay, the next cell in our lymphoid line is our B cells. We only have about 10 to 20% B cells circulating in our peripheral blood. And these help with humoral immunity and produce antibodies. Okay? They are usually short-lived, living only about a few days or maybe even a few weeks. They're derived from the bone marrow. The B cells differentiate into plasma cells when they're antigenically stimulated. And you might want to look on page 87 in your atlas at a picture of a plasma cell. But again, we're on this lymphoid side, and we just talked about the T cells, but the B cells, they can go on and mature into plasma cells. And this is what you're looking at right here. This is a plasma cell. They come from B cells. And on the next slide, we're going to talk about the killer cell. This is the last one that can be derived on the lymphoid side from the lymphocyte. The natural killer cells only make up about 10% of the lymphs. You do not need an antigenic stimulation to function, or for the natural killer cells to function. They are involved in innate immunity. They originate in the bone marrow, and their primary role is to resist bacteria, viruses, and fungi. As far as the lymphocyte's appearance, the nucleus has very clumped chromatin. So you have this very dark, dense-looking nucleus. It's round. It's mononuclear, so it's going to be round. You're not going to have any lobes. The cytoplasm is blue, scant in color, and it's even can be considered sky blue in color. Northern lymphocytes are small and mostly nucleus, so the nucleus is taking up most of the cell and are scant in the amount of cytoplasm they have. So very little cytoplasm, mostly nucleus. And lymphocytes make up about 20 to 40% of normal peripheral white blood cells. This is just a fun little cartoon of the different type of lymphocytes that are out there. You have your B cells. All these little Y's that are hanging off of this B cell are antibodies because they're antibody producing. Then you have your subcategories of your T cells. You have your regular old T cell, and then you have your natural killer cell over here. Next up for our mononuclear cells, we need to discuss monocytes. This is a perfect example of what a monocyte looks like, but we'll get into a little bit more details on the next slides. These are the steps in the monocyte maturation sequence, and you can look in your atlas on page 71 to 77, so you can see a little bit better images. But remember, in your flow chart of the hemopoiesis of all your blood cells, the monocytes are coming down the myeloid side. Okay, the lymphocytes are over on the lymphoid side, but the monocytes are on the myeloid side. So lymphocytes on the lymphoid side, myelocytes are on the myeloid side. So don't get that confused. And look back on page 13 at hemopoiesis at that flowchart if you need to in your atlas. But let's go on and monocyte maturation. The first step is your monoblast. The second step is the promonocyte. And then, of course, you would end up with your mature monocyte. So monocytes, what are their functions? Monocytes leave the bone marrow and in the peripheral blood for a short time in transition to entering the tissues, and they mature into macrophages. Thus, monocytes are young cells, and their main function is phagocytosis. So they only spend a short period of time in circulation because they ultimately want to get to tissues or organs, and they're going to become macrophages. And they're only going to be, like I said, when I say short time, I'm only saying a day or so in circulation. So let's look over here at our diagram. We're going to start here. We have our macrophage. So our monocyte has matured into a macrophage, and you can see there's an infected cell, and this macrophage is engulfing this infected cell. Well, if you remember from one of the previous slides, guess who helps out our macrophages? Our T cells. So our macrophage and our T cells working together to get rid of and kill and take out this cell. So this is the whole process that we go through, or our bodies go through, to ultimately end up and take care of an infected cell or our foreign objects. Those macrophages and those T cells kind of work together in order to take out, phagocytize and take out those foreign or infected cells. So once those monocytes become macrophages and they're in the tissues, they become either fixed or non-fixed macrophages. The fixed macrophages will stay in the same tissue or organ. The non-fixed can wander to different areas. But those fixed macrophages, they become known as certain terms or certain names. So if a fixed macrophage becomes fixed and lives in the liver, those macrophages become known as Kupffer cells. If they become mature and they live in the bone, they become known as osteoclasts. For the skin, they're known as Langerhans. Macrophages in the tissue are called histocytes. Macrophages in the brain and nerve tissue is called microglial cells. So let's look at our appearance of our monocytes, and I encourage you to look at page 75 in your atlas. But our nucleus can be anywhere from round, folded, convoluted, meaning it can have these dips or folds, horseshoe-shaped, so a lot of different variations, irregularities in our nucleus shape for our monocytes. For the chromatin in our nucleus, it's known to be fine and lacy. You'll just notice it's not as dense looking as some of the other cells. For the cytoplasm, it has what's said to be a ground glass appearance. You have this light bluish color, slate gray to bluish color, maybe even purple, but it's usually this slate blue color. You'll have pseudopods, these little arms that can come out because you can have these cells being phagocytic or phagocytized or engulfing cells. You also see vacuoles, all these little things that look almost like air bubbles. These are vacuoles, and again they can phagocytize, so they're phagocytic. Monocytes in the peripheral blood are the largest of your white blood cells. So you see, in comparison to these red blood cells, these are normal shaped red blood cells, how big this guy is. They're also going to be irregular in shape overall as far as the edge or their cell membrane. They only make up about 3 to 8% of your white blood cells in your peripheral blood. This concludes our lecture on mononuclear white blood cells. If you have any questions, don't hesitate to reach out.